MagPix is a compact fluorescent-based detection system capable of simultaneously measuring up to 50 analytes in a single microtiter well. Unlike the Luminex 200 and FlexMap 3D systems, which are based on the principle of flow cytometry, the MagPix system is a fluorescent imager. The system consists of the following components. The system fluidics include the syringe pump, sample probe, and sample valve. These components are accessible through the side access door panel for ease of maintenance. The sample tray is compatible with standard 96-well flat bottom, V-bottom, and round bottom plates. Onboard dry fluid and waste containers include liquid level sensors to alert the user when the system is running dry and to prevent spillage. The heart of the system is the optics module, which includes the sample chamber, magnet, light-emitting diodes, and a CCD camera. Based on Luminex's proven XMAC technology, the MagPix system uses color-coded magnetic microspheres as the substrate on which assays are performed. MagPix can perform quantitative analysis of proteins and nucleic acids in a variety of sample matrices. Here's how it's done. The surfaces of Luminex MagPlex microspheres are easily coupled with a reagent that is specific to the analyte being tested, such as a monoclonal antibody, protein, or DNA sequence. When the coupled microspheres are loaded onto a microtiter plate and mixed with the biological sample, analytes are captured based on specific antibody protein recognition or DNA hybridization. The complex is biotinylated, and finally the detection molecule is attached. The samples are now ready to be read on the MagPix system. A sample probe draws the suspended microsphere-based assay into the instrument. The beads are carried by the system dry fluid into a chamber. As this is happening, a magnet moves into place to hold the microspheres in position. The microspheres settle in a monolayer fashion on the back of the chamber. Red light-emitting diodes, LEDs, illuminate the chamber, and the CCD camera captures a set of images of the fluorescent microspheres. The software processes the images and identifies the beads based on their fluorescent pattern. Green LEDs then illuminate the chamber, and if the analyte of interest is present, the CCD camera will capture an image of the fluorescent detection molecule. The software processes the image and determines the concentration of the analyte of interest from a standard curve. Once the sample has been read, the magnet is moved away from the chamber, allowing the microspheres to be swept out. The system then flushes the chamber in preparation for the next sample. Luminex Exponent software reports the results of the multiplexed assays in real time. In this way, MagPix allows the completion of up to 4,800 individual tests in less than 1 hour. MagPix is a cost-effective, versatile multiplexing platform that provides the researcher a tool that requires less sample, less reagent, and lower labor inputs than traditional testing methods such as ELISA, thereby reducing the cost per data point for testing of multiple analytes. The FlexMap 3D is Luminex's most advanced multiplexed analyzer. Fast read time, high dynamic range, and the ability to simultaneously measure up to 500 genes or proteins from a small sample volume make it our most flexible multiplexing platform. The FlexMap 3D is used in a wide variety of applications, including extracellular protein expression profiling, cell signaling profiling, pathogen detection, vaccine testing, gene expression, SNP genotyping, serology, and HLA typing. Polystyrene beads are at the heart of all Luminex multiplexed assays. These beads are available in a magnetic format, making them easier to handle and automation-friendly. They are internally dyed with precise concentrations of 3 fluorophores, giving each bead set a unique color code. Using this process, we have created 500 unique bead sets. biological reagents specific to the analyte being tested can be coupled to the surface of the beads. This flexibility allows you to perform a variety of different tests, including nucleic acid assays, immunoassays, receptor ligand assays, and enzymatic assays on a single platform. 3 basic types of immunoassays can be performed with XMAP technology: capture sandwich, indirect detection, and competitive inhibition. Here is how a capture sandwich assay is performed. In this type of assay, a monoclonal antibody is coupled to the surface of the Luminex beads. When the coupled beads are loaded onto a microtiter plate and mixed with a biological sample, the protein of interest is captured based on specific antibody protein recognition. The complex is biotinylated, and finally the detection molecule is attached. The samples are now ready to be read on the FlexMap 3D system. 3 basic types of immunoassays can be performed with XMAP technology: capture sandwich, indirect detection, and competitive inhibition. Here is how indirect detection is performed. Detection of specific antibodies in a test sample can be accomplished by an indirect detection method. In this type of assay, an antigen is coupled to the surface of the Luminex beads. When the coupled beads are loaded onto a microtiter plate and mixed with a biological sample, the antigen-specific antibody is captured on the surface of the bead. A biotinylated anti-species antibody is added to the mix, and the complex is labeled with streptavidin phycoerythrin. The samples are now ready to be read on the FlexMap 3D system. It's gene expression products in a multiplexed format using ligation-dependent amplification. In this type of assay, a polyadenylated RNA from total RNA or lysates is captured from solution using DT oligonucleotides and immobilized to a substrate. Following this capture step, cellular debris or other unbound materials are washed away. A complementary DNA strand is then synthesized from the captured RNA, and the RNA is removed. LDA probes for each of the target transcripts are added in a hybridization step. For gene expression measurement, 2 LDA probes are used that are complementary to 2 adjacent 25 base regions of the targeted cDNA. Luminex's proprietary probe design software generates probes that achieve 2 levels of specificity through both non-cross-hybridizing probe sequences and the requirement that both probes must be perfectly juxtaposed in hybridization in order to detect the targeted cDNA. Following hybridization, ligase is used to join the perfectly hybridized upstream and downstream LDA probes together. The joined probes form a template that is then amplified using 2 universal PCR probes on the flanking sequence of all joined probe sets. This approach eliminates both primer and product size biases seen in other multiplex PCR methods. The universal reverse primer is biotinylated for detection in the next step. The resulting biotinylated PCR products each contain a Luminex tag specific for the targeted transcript. The DNA is then incubated with XTAG beads, and tags on the DNA hybridized to their complementary XTAG sequence on specific bead regions. The beads in captured DNA are then incubated with streptavidin phycoerythrin, which binds to the biotins on the DNA. The labeled DNA is ready to be detected on the FlexMap 3D instrument. A sample probe draws the suspended bead with the captured and labeled analyte into the instrument. The beads are carried by the system sheath fluid into a cuvette where the randomly distributed beads line up in a single file. This allows the particles to be measured discretely. As each bead passes through the cuvette, a red laser excites the internal dyes, allowing proper classification of the bead, which in turn identifies the analyte attached to the bead. A green laser excites the labeling molecule bound to the analyte on the bead, providing information on the amount of analyte present. Both classification and reporter readings are made on each individual bead, so the FlexMap 3D system is able to precisely determine multiplexed assay results. Luminex Exponent software processes the data and can be used to analyze the test results. Results obtained with the Luminex software can also be exported to any data analysis program of your choosing. The FlexMap 3D system assures high throughput without compromising flexibility or performance. Optimize your workflow with automated routine tasks, integrated front-end sample preparation, and acquire more answers per test than ever before. FlexMap 3D helps you meet the constant demands for high-quality data with features like fast read times, the ability to simultaneously measure up to 500 genes or proteins, a large menu of commercial kits and reagents to develop custom assays, front-end automation and laboratory information systems integration, compatibility with both 96 and 384 well plate formats, automated startup and shutdown routines, and it's easy to use.
send greetings on behalf of the people of our planet. We step out of our solar system seeking only peace and friendship.
us with a question.